will be happening online. So as long as you have a decent internet connection and a browser and any sort of operating system, Windows, Linux, or Mac, or any sort of devices, mobile, computer, or laptop, or desktop, or tablet, or anything which is kind of have a browser, you can attend it. As all of you have already logged on via an application called Zoom, so we'll be having all our meetings via Zoom, all our classes. These are live classes. So you can raise your hand and ask questions. You can write the questions in the chat box, which Mohit was telling you a few minutes back. So here during this program, so I would request you to mute your microphone so that mute your microphone so that the recording of the session has to be done and will be provided to you for later usage, and so that it is uninterrupted and kind of give you a good understanding of the things that will be covered here. So there will be the people who will be kind of giving you lectures is me, Pratim Chakravarti from Bioinformatics, Dr. Mohit Majumdar, who is also from Bioinformatics, and a few of the sessions will be taken up by Amity University Assistant Professor, Dr. Chito Brotomal. He will also be taking a few sessions with you, and so that all of you who are on site with Amity, you can go forward and ask questions, clarify your queries with Chito Broto as well. He's a pretty helpful guy and also pretty enlightened by doing certain. He has been associated with us for quite a long time, so he knows the in out of the substances which are happening here at Pine Biotech, and also the kind of programs that we are running here and there. So today is the first day. It's called Introduction to Omics Logic. Today we'll be covering the tools and the resources for this program, which will be used by you guys. Can somebody mute his or her microphone, please?
Pratim, can you do the full screen? I mean, it will be more visible then.
Okay. Is that okay, Mohit? Is that okay, Mohit?
Yes, thanks, Pratim.
Okay, welcome. So as I was telling you, today is the first day, July 19th. So as I was telling you that also it's an ice-breaking session to introduce you about the different things that we'll be using, and also the tools and the resources we'll be covering, and what are the expectations, what are the projects which kind of are there and you might be interested in doing at the end of the course. So the course is divided into equivalent mixture of three things: an extensive theory, and extensive practical using the software platform, which I'll be showing you in a few minutes, and also some educational resources, which is a web portal where probably many of you have done your login and hovered around there. And also the structure of the course, and there are some important deadlines which all of us has to meet. As participants, you also have the responsibility to meet those deadlines so that you are learning. So this course will kind of enable you in the domain of biology, where omics data plays a big role, and also trying to understand the biological the logic behind the biology, and also the data aspects of it, which is becoming very important nowadays as a part of biology teaching programs. So the first thing I would like to tell you is about the educational platform that we have. So let me go to the platform so that you can see what's happening here. So if you type this address, https://edu.t-bio.info, will find this page. And on the right hand top corner, you will find a thing called login. If you click on that, there will be a login screen. As you can see already that in my computer, I have saved my name and also the password. And all of you might have already logged on to the Edu platform. If you have not, so I'll give you a few minutes so that you can do the same thing in your own computer. Could you please go forward, all of you, to go to the this particular link. Now I'll paste the link in the chat box so that you can see it. After you have entered your username here and your password here, and then you have to click this I am not a robot. Now this is the security signature which helps you to keep our site safe, and you will find a visual. So you select all this stuff and you do a verify. We'll say that I am not a robot will be set at green, which means that this has approved your login. Then you log in. A message will appear in the bottom of the login that login successful, redirecting. And then you will go into the page. Now this is the page that I can see because all of you will be seeing the same screen. The only difference between you and me would be this photo and the name, and also something which is called points. I will tell you all about this, about the points later on, but first make sure that you are able to log on. Have you guys even been able to log on, all of you? I'll give you a few minutes so that all of you can log on.
Yes, if you have any issues with login, you can always write it in the chat box. Apar here is an intern with us. Subrajit is also an intern with us. They'll be also helping you guys with any login related issues. So please log in.
So I guess all of you have been able to log on. So here you will find, so now you're in the home. As you can see, underlined, the underlined thing out here in the menu tells you where you are. So if you go to the courses, you will find an array of things out here. On the left panel, you can see the various educational courses that we have. So we will be covering whatever things that we'll be covering in this course for 3 months. A detailed practice of it should be there, and that's why we have created this thing. And all of you can go there and finish the course and do the course on your own accord or your own pace, so that you are in pace with the various things that we are going to cover during this session for 3 months. Now, the The first introduction to bioinformatics is the flagship course. So let us go out there. Let us see what we have here. So this is the course for introduction to bioinformatics. Now there are 4 tabs: overview, curriculum, instructor, and reviews. Overview tells you about the course description. Okay, it tells you about what all are there in the course. There is a small video out here, educational video, which you can always play at your own end and find out that what all is there in the entire edu.to-bio.info, edu.t-bio.info platform. In the next step, there is something called curriculum. The curriculum tells you more granular details about the stuff. So it starts with an introduction survey. The survey asks you some questions which will help us and help yourself also to understand that what level of understanding you are, and also will give you a pointer how much do you need to really learn, your level of understanding, and also it will tell you all those things that you need to do to upskill yourself and to go forward in learning the things that are there in this website. And also you can also go to some other places. This will give you the pointers about the introduction to bioinformatics. So what do you actually need to know before embarking on the thing? Even if you don't know, that's okay, but you will know that what do you need to know, so that you can get back to your books, get back to your professors, get back to your teachers in the colleges, in the universities, and get to know the things about it. The third tab is the instructor, and she is the lady who has developed this course. And the final one is the review. So as you can see that many people have done the course and written their review. You can find your fellow colleagues in your university. Shoham was a part of this program and has done this course, Introduction to Bioinformatics. We also have Shubhrajit. Shubhrajit is a research intern with us for the last 1 month, and he's kind of doing some projects with us, and he has a good understanding. So you can always go back to Shubhrajit and Shoham in the university if you have any things to ask. I mean, you can always reach out to us as well, but since you are friends, you can also get back to them. Now, please complete this course. Once you complete this course, I would request you to write a review of your understanding. Did you feel about this course details, etc. And please do not forget to mention a title, that one-liner. You may say it's bad and it's not worth doing or something like that. You are always free to do that as well. And if you like it, but you should also mention it that, okay, I liked it. Besides Introduction to Bioinformatics, there are other courses as well. For example, Transcriptomics 1.
Pratim, can you show
Yes.
us the certification that one would get after completion of the course?
Yeah, of course. Yes, of course. So I missed out one thing, guys. So this is like, once you are done with the course, for example, as you can see, all of the modules out here are, there's a green mark, which means that I have completed all these things. So after the completion of the course, you get a certificate. If you click on that, a certificate will be generated. Sorry. Let us go to Transcriptomics 1 and find it out. Okay, so I have completed this thing. Oops. So something is happening at my end. So maybe it's not working for me. So I'll show you. I will show you about the certification part. I already have a certificate in the presentation itself. I'll show it to you. Don't worry. Okay, so going back to the course details, you have transcriptomics 1 here. You have, and as you can see, you know, all the modules that I have completed so that it is marked green. So when you complete the courses, it will be marked as green. And the very interesting part of this course is that you have a quiz at the end of each set of modules. The quiz are kind of benchmarks. They will help you to understand that what are the common questions that you need to understand, what are the common concepts that you need to know, and that will help you to understand how much you have learned and what remains to be learned. Going forward, there are other courses as well. Transcriptomics 2, Transcriptomics 3, Genomics 1, Metagenomics 1. We'll be covering transcriptomics, metagenomics, all these things in our lectures. But at the same time, all of you have to go to these places to have a deeper understanding of the things. That what does it really mean? How does it really happen? So, and also practical exercises are there in the modules itself, which will tell you about the actual data analysis that is being done in the metagenomics domain or in the transcriptomics domain or in the genomics domain or anything else, or epigenomics, something like that. So there are other places too. There's a tab called events. If you click on that, it will tell you about all the upcoming events that are occurring across the globe and are conducted by Pine Biotech. For example, as you can see that the next lecture that is going to happen on Pine Biotech event, as a part of Pine Biotech event or Amity University event, is this introduction to omics logic on 22nd of July. And we also have a precision oncology lecture which is going to come up in the 22nd of July program. But since you are a part, I mean, the people who are right now here is a part of this omics logic program, you can attend this always. You also have a number of projects which are reproduced from peer-reviewed papers, publications, and the data that are there available in this publication has been used to create a project for each of you. During the entire length of the course, you will get access to all the projects that are there. I mean, you can always go to that particular project, do the project at your own end, ask questions to us. For example, as you can see that we have projects on Spodoptera, Ebola, and crop dot stress, targeted medicine, and so on and so forth. Neurodegenerative disorders, analysis of changing immune response in cancer, patient-derived xenografts model. Now, these concepts are very important in the industry and also in the academia nowadays, where biologists who are generating data from a number of high-throughput experiments are, using the data science, bioinformatics to analyze and come up with a biological insight using computational tools. So these are some of the projects that are there. During the entire length of the course, you will have access to all these projects. Feel free and find your time to go into this website and find out about these projects and do it by yourself so that you can get an understanding about how the data analysis happens in the domain of bioinformatics. And this will also help you to understand the actual scenario of the research and also the industry needs, and will definitely make you more employable to the industry, and also make you more able in doing your research if you are trying to address a research career. There's a blog out here. So the blog tells you about the events, about the different things that we are kind of doing across the globe. For example, this is one of the program, Omics Logic Online Genomics Training Program that we're going to ensue. This is another program. You are a part of this Amity University Summer Bioinformatics program. So you will get all the updates of whatever is happening at Pine Biotech in the page. And definitely, you will mm, get back to us. You can always get back to us, me, Mohit, Apar, and Shubhrajit, to know about the things and also ask questions in the forum itself, in the chat itself while the lecture is going on. And also, you can, if you're a part of the WhatsApp group that we have already made, you can ask questions and get your questions answered by us. So going forward to the presentation. So these are the courses that we saw, and we will be covering in our 3-month lecture. We'll be covering bioinformatics, the transcriptomics, the metagenomics, and definitely machine learning. But at the same time, you are free to go, as I showed you, you are free to go to the educational website. If you have this presentation already, you can click on this link, which is at the header, and you can directly go into the website and find it out. So this is the organizational page I'm talking about. And now let us go out there. So if you go below to the bottom of the things, you can find something called organizations. Okay. Go to Amity University, click on it. See, this is the, I'm pasting this link in the chat box so that all of you can see. So if you open this link, you will find several tabs out here as always. Home, forum, progress, curriculum, assignments, and courses. Now this is the timeline. Okay. So today is 19th July. If you click on that, we are on the first day of the course, which says Introduction to Omics Logic. It's an online course, and what all will be covered. So individual programs can be registered. So don't worry, you have already registered for the entire course. So you can find the timeline here. So if you click on all the dates, you can find what all we will be covering in each of the dates. So from 19 July, between 19 July to 26 July, we'll be covering Introduction to Omics Logic. From 26 July, we'll be covering high throughput data and NGS data analysis. So between 19 to 26, we'll have, more sessions, 3 sessions to be very specific. So today is 19, the next session is on 22nd, and the next session would be on 26th. From 26th, we start with high throughput data. So these are the broad heads, broad topics that we have mentioned out here. If you go to 5th August, you will find that this is the start date for doing the transcriptomics. And 19th August would be like, metagenomics and microbiome. We'll be studying all the things about it. So, and then to 2nd September and going forward, we'll be, we'll be starting to learn on machine learning from 2nd of September. And from 9th September onwards, we'll be addressing the bioinformatic projects. So for example, this is the forum page. This is the forum page where you can paste your query. For example, as you can see that Shubhrajit has joined, Chittabrata has joined, Gunjan Sharma has joined, and many other people. So I would request people like this, BC8, 89, GOU, Arabi, Gaurav. I think, so could you please, write your name so that we know each other. We are also trying to create a forum here, forum of common interests, people with common interests and people with common understanding. And therefore they can, maybe they can collaborate between themselves in the future and discuss stuff like that. So I would also, I would also request this guy, Enray, who has joined this group to write the full name. For example, Jito Proto is, has joined with a name and also a photo. And I will tell you about it. So many of you I can see have joined, uh, all these people. So going forward, let us go to the forum. So this is the place where you can find, discussions. You can find, for example, the first topic that has been covered out here has been given by Mohit. Mohit has pasted the meeting link out here. So please be noted that this meeting link will be used for the entire length of the program, which is starting today. So you don't have to, kind of grope around in your email or anywhere else or ask anybody, but you can just simply go to the forum and find the link to join. So you can, for example, you can reply to this post, or you can make a new forum for yourself, new discussion forum for yourself, and and gradually go forward. So if I go to the forum again, you can see a place where create a new topic on Amity University Bioinformatics training program. Now, if you have any question, you can write the question here, something like this, okay? And please give a title to your query. For example, I need to understand the concepts of machine learning, let's say. Or let's say, which resources do you suggest for understanding the, pardon my spelling mistakes, machine learning. And you can write your details about it. And then you can say, typically we keep it normal, and then topic status would be open. So you want people to answer you back. So this is a very active forum. In other programs that we have, we have a very active forum where we talk about stuff, we exchange stuff, we exchange publications, news, and everything out here. And you can always go forward to discuss between yourselves. So this is an e-learning forum. So all of you can access it. All of you can post it out here. All of you can understand what other people are thinking and other people are doing at the same time. So please feel free to post your stuff. That will make a collaborative learning much better. Going forward to the progress. Here you can note down you can see your progress, your own progress in this particular program. For example, as you can see that these are the people who are already logged on to the server. So this is the educational platform which I'm showing to you right now. There is also a server. There's also a software platform. For example, as you can see, Shubhrajit has been using quite a lot of server nowadays as a part of his research internship. Now he has scored. So all of you, your score will be coming here. So since all of you have already joined this program, the default marks that you have already got is 100. So I can see a number of people out here, but we have almost 60 people. So I would request all of you to log on to the educational platform, and so that you get a marks out here, and we all know that all of you are here, and this will be a very more the merrier. So please be free to log on and make your profile here. And this graph will show you the progress. For example, this is the thing that pertaining to the Amity University. So for example, this will show you, this graph will show you whatever is, today is July 19. See, so this graph shows that how much server has been used. So in this particular program, since Shubhrojit is the only person who has used a lot of server, this graph is pertaining to Shubhrojit only. But as you people who are, rest of the people who are there, who will be using a lot of software platform, we want to see this yellow graph. This yellow mark graph will point to the fact that you people are accessing the educational platform and also the server as well. And during the entire length of the course, even if some events are taking place across the globe, somewhere online program somewhere is taking place, all of you will be eligible to be a part of this program. But since you are a part of this Amity program, you will be given a discount for attending that program. Even if it is happening in different parts of the globe, you can attend it, of course, with a discount. So going forward, this is the curriculum page. The curriculum again will tell you about what all we are going to cover here. So this is the timeline. So today is the first day. Although we wanted your Vice Chancellor, your Honorable Vice Chancellor, to be a part of this program, he was very busy, so he could not attend it. But he has given you extended thanks and regards to all of the participants here, particularly from Amity, to be a part of this this thing. So this is the timeline. So if you scroll down, you can find that what all will be there in which date and what are the things that will be covered, so that this also gives you an idea that before coming to class, what kind of prerequisites, or not exactly prerequisites, what will all be covered, so you are becoming aware of this. Any question you have? I think somebody unmuted their microphone. Please ask. Okay, don't worry. So yeah, so for example, if you go down, you can find all these the topics, the broad heads which are being covered out here, and you can find all the details of the course which will be happening every day. Okay, so moving forward, going to the assignments. So if any assignment is given to you, this will be reflected in this page. So since we do not have any assignment here, so as you are going on with the course, when it comes to the part where we are will be doing a lot of project, then this will be appearing here. Now for the time being, this is empty. So each of you has to go into this page. For example, each of you will be having a page like this, empty page like this. So you can at the end of the course, for the last month of the course, will be projects. So you can submit your own project, or you can do the projects that I have shown you, which are there in the projects tab. And then this is the assignment which you have to do at the end of the project. You can fill it up as you go on, not right now, but we will tell you when to fill it up. The courses tab. The courses tab will, these are the courses that are available. See, I talked about all these things. I talked about introduction to bioinformatics. I talked about transcriptomics 1, transcriptomics 2, transcriptomics 3, machine learning, introduction to metagenomics, introduction to metagenomics 1, and so on. So many of you would be kind of inquiring that what is this yellow line? This means that since you are looking at my profile, I have completed all these courses: transcriptomics 1, 2, 3, and introduction to bioinformatics, and 54.55% of metagenomics 1. So that's why it is showing this is a progress bar. So as you people are always going on to do your courses in the educational platform, this sort of bar, as you can see, this course is 100%, which means that I have done it. So I would request you, if you want such a thing to appear in your profile, please go forward to complete the courses. It's very easy, it's very engrossing, it's very interesting, I must say, and this will definitely give you a lot of edge over others to secure a position in the industry, and also secure a position, have a good research career, and also have a good industrial career as well. Okay, so we are done here. This is the organizational page that I wanted to show you. So I think I covered most of it. So I would request you, all of you, between this today and Monday, when is the next lecture day, please go to this website and figure it out. If you have any problems, we can always please get back to us. We can help you accordingly. So these are the projects I was talking to you about. So we have this in the educational platform. So and all these projects, as I was telling you, all these projects will be available to you for study during the entire length of the 3-month program. So our intention of doing this introduction to bioinformatics program with Amity is to give you a project-based education, which means that from the beginning of the courses, we'll be giving you ideas about real-life problems, real-life data sets, and that will be a part of your curriculum, that will be a part of your understanding, and you will get a lot of chance to address these problems using the software platform that I'm going to show you a few minutes later. And there'll be a lot of hands-on here. As I was telling you, 3 different sections: theoretical part, hands-on exercises, and also independent thinking, which is a project part. And this is a wholesome research experience that you'll be having at the end of this entire length of 3 months course. So we plan to, the research projects which we have done are, curated from, peer-reviewed publications across the globe from very prestigious journals. This is a very prestigious journal, Genome Biology, and this article has been curated. The important data sets from this article has been curated to make it into a workable, into a workable data, and using it in the software platform that we have, and coming up with an understanding of the research question and also the biological insights connected to this particular therapeutic area, which is breast cancer out here. So this is a this is a poster session that was held in the Louisiana Biomedical Research Network. This was a part of that, and similar things will obviously happen at Amity. So I would request you to work very hard and be initiated about the program, and kind of we can also organize similar stuff at your university. That's what the plan is. Now this is the software platform that I was telling you about. It's called server.t-bio.info. So if you have the slide, you can click on this. You can click on this particular link. This will automatically open into a browser. Since I already have the browser opened in my computer, I can show it to you.
And then we will try and visualize the samples based on all.
Pardon me. Okay, so this is the software platform. If you can see my screen properly, let me maximize my screen. Yes, so this is it. So since I have already logged on, I can see this. Okay. Now, if I want to log out, I have to kind of log out. Signed out successfully. Let's log in here. So I already have typed my password. So if you have this password with you to log into the server, I would request all of you, I will take a 5 minutes gap, and I would request all of you to log in into the server and let us know whether you are able to do it. Okay, so yes. So let us paste the server link here. Please log on to this web page. I guess most of you will be able to do it. Now, please remember that many of you were complaining about not being able to log on because the password is some case sensitive, and it is a password generated. It's a generated password. So please be very sure about the cases and also some numbers. So if you are not being able to log on, please let us know. Now, this is very important because, during the entire length of the course, we'll be using a lot of this software platform. So it is very important that you are able to log on from the first day itself, so that as we go on into the next lectures, as we go on learning about transcriptomics, metagenomics, and all this stuff, this software platform will be used highly by you guys, and this software platform, the algorithms, the softwares that are the pipelines that are there in this particular software platform will be used extensively by you people to understand about the typical data analysis that takes place in the bioinformatics domain using the data science principles. So all of you, I think, Akash, you are not being able to log on. Okay, so somebody says, I have forgotten my password. You have been given the password over email. Can you check your email? Ritik says it still shows as invalid. Arijit, you cannot log in. Okay, so all of you kind of having this problem that it is showing invalid. Apart, Anushka, there are no many people. Shubha Lakshmi, Madhavendra. Okay, we'll take care of it. Shayantoni.
Yeah, I think
Vinaya.
Sahil is here, I guess.
Sahil, can you just pitch in here to help us out? Sahil?
Yeah, sure. I mean, I got it, and I can send out the email for whosoever who's not able to log in.
Yeah.
Yeah, I'm gonna send out the email.
Most of the people are kind of having a problem.
Sure.
It's being invalid. So
Sure.
So don't worry, guys. Sahil will take care of it, and a mail will be sent to you people. And during this time from today to Monday, you'll be having this email, and then you can check on yourself. Do not worry. Please do not worry. It happens sometimes. After all, it's no. These are machines. So don't worry about it. We will take care, and we will handle it. Okay, don't worry, not worry. No, I mean, I can see people are kind of frantically writing messages, so don't worry. Yeah, Shoham, you have already logged on earlier, so okay, so yeah, so please log out, Shoham. Then you can always check that whether your password is also working properly or not. So do not worry, you guys. So do not panic. We will take care of it. And definitely, you'll be given the right credentials. And if you are having problems, still having problem, please get back to us. We'll sort it out. So we have enough time in our hand. Just listen to what I'm saying. Just watch the demonstration, and then it would be fine for you guys. Sir, I logged in. Is it showing or not? So if you are able to log in, Elena, then you will have a, let's log in. You will find this green bar on the top which says signed in successfully. So if you're able to see it, then you are more than sure that you have logged on. Okay, so yeah, this is the software platform that we'll be using a lot, and this has several sections like NGS data analysis. And if you go to each of these, this top capital lettered headings, you will see a pop-up. This we call this educational pop-ups. So NGS data. So it will tell you all about next generation sequencing. So it will tell you a brief about next generation sequencing. What is it? You can read it. You can so this thing will give you a broad overview of the things that are happening in the next generation sequencing world. So if you go here, it will tell you about mass spectroscopy. So what is mass spectroscopy? And there's an interesting visual out here. So you read it. So these are just pointers, okay? So you can have a very broad overview. Since you people are students, you people are just starting off, it is very important for you guys to understand the basic concepts of it. So this is just a pointer. This is just a very brief paragraph on mass spectroscopy. So read it. You will find it definitely very interesting. Then there is something, we have a modules in structural biology. So structural biology is a very interesting subject where protein structure and which kind of dictates protein functions are covered in this domain. So this again gives you a overview of the stuff. And then this is the machine learning part, the data integration and the modeling. This is mostly machine learning. So and each of these sections like multiomics and all these things will give you ideas about the various concepts of machine learning that are going to be addressed during the course. Okay, so moving on, I think we are yes. So if you log on, you can later on you can find something like this. Do not worry about this visual. This is something you will be kind of getting accustomed very soon, and you'll be doing a lot of analysis using this particular software platform. And I'm sure with the initiation that you guys have, you will have a lot of understanding about how the data analysis takes place, what kind of data is used as input, what kind of parameters that it uses. I mean, it might seem a little, difficult words when I speak, but be very sure that, we'll teach you all this stuff. And once you're logged on into the server, you can see something like this and you'll have a great time. I can guarantee that. Okay, so what Mohit was telling you about certificates. So a successful completion of this course will give you a certificate, which you can see on the left of your screen, where it will be a certificate from Amity University and Pine Biotech collaboratively. And you can, this will be you can post it up in your social media page. You can include that in your, your CV. You can, this will definitely tell you that whatever you have done, what all you have done. So again, there's a question out here. I think, many people have asked me personally that we need to have, what will ensure that I get a certificate? So for example, as you can see, this is the certificate that I was telling you about when you complete a course. For example, your professor, Chitobroto, has completed Introduction to Bioinformatics, Transcriptomics 1, and Transcriptomics 1 and Transcriptomics 2. So these are some of the certificates that are generated by the educational platform itself. So when you complete one educational course in the educational platform, let's say you complete Introduction to Bioinformatics, you will get a certificate like this. If you complete Transcriptomics 1, you'll get a certificate like this, wherein your name will be written. Okay, if you complete transcriptomics 2, you will also get a certificate like that, which you can obviously share in the social media. You can always share, in your CV. You can always tell people that, okay, you have done this. And mind it, this certificate, which you can see on the left, it is an internationally valid certificate. So if you go anywhere and say that you have done a program which has been jointly conducted by Amity University and Pine Biotech, I mean, this certificate is valid across the globe. Okay, so the next session will be on big data bioinformatics, where we'll be covering a lot of how the data, biological data, is being looked upon, and we'll talk about the examples in the next session. And so you can always in the meantime, you try to log on to the server, you kind of log on to the educational platform and hover around there, play around there, just kind of, have a feel of things, what are happening. So this will kind of help you to understand that what are the things that we are going to cover. I will quickly go through each of the slides, next few slides, because this will tell you about what are the things that are coming up as a part of this course. On July 26th, we are talking about the high throughput experiments. And here we will cover a lot of, basic molecular biology, like the central dogma and the DNA code and the transcription and the translation. And the most important part about biological data understanding is the quantification. And the quantification takes place at the molecular level and also at the subcellular level. And at the same time, we will also be talking about the next generation sequencing, particularly for the biomolecules of DNA and the RNA. And here you will understand about the technologies that are used across the globe to understand the gene expressions, to quantify the gene expressions, and what all we can do to have a biological insight using this next generation sequencing data. So this session will start from July 26th. The next session is on July 29th. This will be taken up by Chitobroto. I would request Chitobroto to take the stage and kind of give you some introduction about him, a small introduction about himself, and also tell about this Transcriptomics 1 program. Chitobroto?
Thank you, Dr. Pratim.
Yeah, Chitobroto, could you speak a little louder?
Thank you, Dr. Pratim, for introducing me. So, Amity people should know me as I am taking their classes there right now. Many of them know me very well. And apart from Amity University, for rest, I am the assistant professor there. So, as already Pratim mentioned, so I will take these two sessions, T1 and T2, where I will cover the next generation sequencing and then the analysis of variants. In the first lecture, next generation sequencing, I will introduce the RNA-seq pipeline to find out the differentially expressed genes, and before that, what we need to do for the data processing, and that is known as pre-processing steps. Okay, and then different kinds of mapping the RNA-seq to the transcriptome or genome. We'll see, we'll look into that, and after that we'll study the quantification techniques of the RNA-seq and the isoform. And in the next class, that is on August 5, we'll look into the data normalization part, the filtering steps to remove the noise is there, because the RNA-seq data is a large scale data and there will be lots of noise. You should remove the noise, and then to extract the meaningful information from the large scale data set. That means you are using multi-dimensional data sets, and from that you need to identify your your gene of interest, which genes are expressed more or which genes are expressed less in quantity, so that you can analyze in using these pipelines. And then obviously the principal component analysis, which is a statistical technique, and we will see how we can do that using this Pine Biotech pipelines, that means this T-BioInfo server. Thank you all.
Thank you, Chitra Brother, for your excellent introduction. So people, you will learn a lot. And since Chitra Brother is available in the university itself, so he can help you to go forward and understanding the various concepts. And accordingly, he will definitely be using a lot of pipelines and also the educational platform to do the courses. The next session will be on August 12th. It will be on gene expression analysis. And as Chitra Brother will be already introducing the basics of machine learning out here, we'll be kind of taking it forward to understand the, when it comes to gene expression, it is very important to understand the correlations of between the features and the factors. Do not be scared about the words like features and factors. We'll be explaining all these things to you, and these are the kind of buzzwords or the keywords that are typically used in the data science pertaining to biological data. Okay, we'll also learn about the clustering. Clustering is nothing but trying to understand about how the kind of how the gene expressions group together, and also kind of creating profiles out of it. The profile, by profile I mean that something by which a particular gene is identified across the entire pan-genome thing. Okay, so we have a gene pool. So how the genes together function together. What are the genes that correlate? What are the genes that cluster together while they're kind of expressed in the mRNA level and produce the right kind of protein so that it is participating in the in your biological activities inside your body, inside the body of organisms. And we'll also talk about the expression profiles and across samples. By samples, I mean whatever it is meant by the sample is nothing but the various therapeutic areas, to be very specific, like let's say cancer, or let's say tuberculosis, or let's say some other infectious disease. How the expression of the genes in the human body, how the expression of the genes in the pathogenic organisms, and also the host, and also the infectious the viruses, the bacteria, etc., go forward to kind of produce the detrimental effect within the body itself. So we will talk about this in the gene expression analysis T3, which is going to be held on August 12th. The next session will be on August 19th, which is on metagenomics. Metagenomes are something which is kind of organisms which are very, it is present across the globe in various, you can find it in the soil, you can find it in many of the extreme climates, let's say in hot springs or very cold, minus 42 or minus 45 degrees centigrade, you can find bacteria surviving, they're surviving, they're just chilling out there, and they're just kind of doing all the metabolic functions very easily. Now, what do the genomes tell us? I mean, very interesting, because they have been there since time immemorial. They have been there when the earth was actually, a boiling pool of magma. But how did they survive? How did they kind of, in the evolution cycle, these metagenomic organisms plays a big role. So we'll be studying about it. And also we'll study about the microbiomes, because microbiomes also plays a big role, as you all know, or maybe you are aware of, that we humans have a lot of microbiomes in our stomach, which helps us to digest the food. So we'll be talking about that, and we'll be talking about the next generation sequencing technologies pertaining to the metagenomics, and we'll be talking about the 16S hypervariable region. So 16S hypervariable regions are something which differentiates between the various metagenomes. And then we'll talk about the gut microbiota, which I was talking a few minutes back. and also the connection with the human health, that how the gut microbiome plays a very interesting role, a very sweet role to control the various metabolic functions inside our body. And then we'll go into the metagenomics one, which is a little deeper, and the research areas right now in the domain of metagenomics. And now we'll kind of use the pipeline, the software pipeline that I showed you. We will use our data to analyze the metagenomics, metagenomes to understand the gene expressions and all these things. And we will definitely, not only we will analyze, we will also interpret the data. What is the biological interpretation? What does it mean really in the biological parlance? So this we will be covering a lot in the Metagenomics 1 module, which will be held on August 26th. Then we will go into the machine learning, the buzzword of the decade. And I would request Mohit to kind of give some introduction about himself, who is also a faculty in this program, and he will tell you a little bit about it. Mohit, the stage is all yours.
Yeah, Pratim, thank you. So yeah, machine learning, as you all know, is a buzzword, and it's been used.
Mohit, could you speak a little louder? It's a little less audible.
Okay. Yeah. So I was saying, welcome to you all, and before jumping into all these topics, one should understand, or it's based on my personal experience, that we are covering a lot of things during this 3 months course. These are very broad topics that we are covering. So definitely will not expect, or you should not expect yourself also to become an expert overnight. You can only become an expert if you go through the educational material. So we have these courses designed based on the educational material that is there in the platform, the educational site that Pratim showed you guys. There you will find all these courses. You click on these courses, you go inside the courses, and you check it out. Page by page, everything is shown in very clear manner. And at the same time, in the next tab, you can open the server, and you can just alt tab, or just switch between the tabs and see how to do it. It's also a manual to use the platform. So the expertise can only come through experience. There is no other way to it. So for that, I would all of you to I would recommend all of you to use the educational and the server platform, because you all guys are here. You guys all have paid for this course, and to make the most, use the materials. And I would say it's very easy. I mean, the way I have used it, or the way I've seen other platforms or whatever is available, and based on my personal experience and people who have been doing it, it's very easy. It's not that difficult, because everything is written there. You just have to read and try. So once you do that, then we can jump into things where we can design things on your own. So that would be a very good experience. Coming back to the topic, introduction to machine learning. Before this, we will be covering, as you know, transcriptomics, metagenomics, introduction to bioinformatics. We'll be talking a lot about data. So what kind of data we'll be talking about? This is very big data. I mean, by big, you cannot even visualize it. It's like that big. So it is in terabytes and megabytes. But a movie, when you download a movie, it's like 1 GB per se. But that's something that you can totally visualize, that okay, this is the movie, this is the video file, that's why it's taking so much space. But what I'm talking about here is a text file. A text file of that size contains huge number of information. The information written in letters. So it's literally impossible. I mean, you cannot open this into a Excel sheet. It's so big. So what to do? I mean, how do we analyze this data? So we have special methods to analyze this data. And nowadays, from mobile technology to any other technology, you see data is generated like humongous. Like even this recording, many people are using Zoom right now, and recording their own meetings. So the amount of data is being generated in everyday basis is so big that it's really, we don't have resources or we don't have minds, you know, to analyze that data, or algorithms perhaps to analyze those data sets. So because of that, you have seen this huge boom in data science. Why this data science has come up? The reason is generation of data. The amount of data and the rate of data generation has increased so much that it's now uncontrollable. You know, it's very important to analyze the data and come to conclusions. That is helping companies, that is helping the government to take good decisions, that is helping everyone. So that's why artificial intelligence and machine learning has become such huge topics. These are topics that includes a lot of statistics and mathematics, but don't get afraid. The pipelines we use is an automation of all these things. We'll be talking about the algorithms, we'll be talking about the data and everything in a very simplistic manner so that everyone can understand. So in this lecture, we'll be talking about the methods such as supervised and unsupervised machine learning methods.
Mohit, you are not audible.
Okay, such as supervised and unsupervised machine learning methods, different statistical methods, regression we'll use factor analysis, we'll see the features from the data, things like that. And we'll also see how these things work on a real data set, like a cancer data set, like what patient samples, their patient samples that have been, you know, recorded in the websites, in the databases. We'll take out those sequences and we'll look at, you know, how we can analyze and come to machine learning and do make understanding out of it. So those are the things that we'll cover in introduction to machine learning. Can you jump to the next slide, Pratham? Hello?
Yeah,
Hello?
I have clicked. Yes, I can. Can you see it? Machine Learning 1.
Yeah. I think I have some internet issues here.
Okay, I have jumped into the next slide.
Thank you. So yeah, in
Yeah.
machine learning 1, in the next week, we'll be talking about the different methods like support vector machines, which are also a part of supervised machine learning methods. And then we'll look into features which are important to make, you know, meaning out of the data. So how can we classify data into, you know, modeling something which will make sense, like, you know, make predictions, do predictive analysis, things like that. So we'll be talking about those methods such as PCA in a very simplistic manner so that you all can understand. And you can always ask queries, whatever questions you have, you can email us. We'll be covering all those things. So in this, we'll also talk about some visualization methods. So it will be a very thrilling journey to learn the data science from perspective of, you know, multi-omics data, and then understand machine learning to how to analyze that data. So it's sort of a project. It's this complete experience will be a very nice experience. Thank you.
Thank you, Mohit. Thank you very much for your, for you giving that understanding. So I can see many people has had wrong Zoom links. So I think this part has been resolved right now. And some of you has been asking me about 80% attendance. So it is always wise, you know, to attend the lectures live so that you can, you know, we can discuss like this, the way we are doing here. And you can ask questions, discuss among ourselves. And you can always, you know, you will be getting the recording of these sessions in the forum itself. But, you know, but it is always good to attend the classes, you know, and because sometimes, you know, it helps to get a feeling that you are in a class itself. So I would request you to, you know, make time. That's why we have kind of shifted the time from 8 p.m. to 9 p.m. towards the night, so that all of you can get back home, settle down with a cup of coffee, maybe, you know, or maybe a cup of tea, or maybe with a glass of juice, maybe, and with a sandwich, and or have your dinner and then sit for this particular session. And definitely, you know, at the same time, I would also request you that, you know, attend the lectures, is only a part of it. But if you really have to kind of understand, or really have to understand how the data is analyzed, and also, how the nitty-gritties of transcriptomics or metagenomics or machine learning or bioinformatics work, you have to kind of go into the educational site and do the courses by yourself. Now, doing the course is always advantageous for each of you. Doing the course will enable you to have the question. I mean, I can guarantee that all of you people kind of are a little scared about, about going into the educational platform and kind of whether you'll be able to understand it and who is going to address the question, that if that is occurring in your mind. Please do not worry. I mean, me, Mohit, Chittobroto are always there to answer your queries, and we are available via email, we are available via WhatsApp, we are available via any form of communication, that is Skype or something like that, or while we'll be kind of organizing workshops in the Amity University itself. 3 workshops, I'm talking about that. Now let us go and kind of figure out. Okay, on September 9th, from September 9th onwards, we'll be starting to kind of frame the bioinformatics project. Now, all that you have learned for this entire length of 2 months about metagenomics data, about transcriptomics data, now let's do a project. Now let's do something which is translational, something which is kind of give you an understanding of how much have you learned. Now, this is not an exam. This is something which will kind of give you that experience about how a research is done. This will give you an experience how things happen in the industry. Now, when it comes to the industry, pharmaceutical industries or the biotech industries, they do a lot of research at the same time. It's not like that it's a job. Yeah, obviously it's a job. You get paid at the end of the month, but at the same time, you have to kind of understand that, what are the kind of things that are happening. So this project will enable you to have that research experience, be it industry, be it academia. So typically it will be a team project, and I think there are so many people across Amity who are kind of attending the program. You can always collaborate with each other. You can have very novel ideas. You can even emulate the project that is there in the educational platform, or always you can kind of devise a research project on yourself. Now, that is always welcome. But it's very important to the project and get a we are planning to do a small seminar by the end of this course, so that all of you can post your research finding as a poster, or as a kind of give a lecture on a particular project that you do. So that will also improve your communication skills. So it's a holistic experience at the end of the day. And we'll be helping you. Don't worry. I mean, we'll all be helping you to do the project. Chittobroto will be there to help you out with the project. We'll be there to help you out with the projects, etc. So during this entire length of 3 months, we'll be conducting workshops. Okay. So for the entire length of 3 months, we'll be having 3 workshops, each 1 month. In a month, each in a month. So the first workshop that is upcoming is on July 30th. So probably for the Amity people, you already have this date in your academic calendar. So if you are already in access with the academic calendar, you can see this date and kind of attend it. It will be happening in a time when many of you have asked me across WhatsApp that do I have to kind of bunk a class and do it? No, not at all. So the workshop will be happening in a time when which is congenial for all of you. Don't have to bunk a class and come over to do the workshop, something like that. So don't you worry. And also people who will not be able to attend the workshop, it will be kind of we will be probably we'll be streaming it the way we are streaming it today. So anybody who who may not be able to come down to the Amity premises in Rajarhat, they can attend it online as well. The second workshop will be held on August the 23rd, where we will be covering we'll be answering your questions. By August 23rd, many of the transcriptomics will be done. So you will definitely have a lot of questions in your mind how to go forward and do it. So we'll be attending those questions, we'll be answering those questions, we'll be having discussions out there, and we will show you that how the things happen in the server, how the things happen in the educational platform, and therefore you will be able to, you'll be more comfortable in going forward. The third workshop will be held on September 16th. Now, this workshop is primarily for the planning of the bioinformatics project that you're going to do. So this requires, here we kind of team you people up, divide you into teams, small teams, or maybe moderately big teams, moderately larger teams, and then you'll be given a topic to discuss. You'll be topic to kind of do the project, and for the next 1 month from September 16th, you will be kind of doing this bioinformatics project. Now, coming back to the educational platform once again. So I would request all of you to make a good profile of yourself in the educational platform so that each of us know each other better. For example, on the left, you can see Apar. Apar Agrawal is a research intern with us. Sorry? You mute your microphone. Somebody who has unmuted it. Could you mute your microphone, please? Anybody who has kind of unmuted it? Thank you. Yes, so, I'm talking about the profile in the educational platform. So if you click on that, you can go to Apar's profile. He has a photo, very nice photo, very smart. So he also has added his description about himself, that what all he's kind of interested, what all he's doing, and he also has added some social links, and this will also add points to your progress. Okay, and on the right there's another Vok Tawe. She's a assistant professor in South Africa, in one of the most prestigious universities in South Africa. So she has done very good job. She has done kind of is a very active member in the educational platform, and by giving yourself a photo and about your description will enable others to know about you. Let's say somebody wants to talk to you, somebody wants to kind of communicate with you. Now it's very important, for example, in Facebook, any social media, this is also a forum, this is also a social media, but restricted for people who have a common sense of understanding, who are trying to learn bioinformatics, and who are also trying to learn the data science aspects of bioinformatics as well. It's very important to have good profiles. So as you go forward by doing the course, build up your profile. As you can see, Apar has substantial number of points because he has done many projects. He has done all the courses. He has done many of the projects that are available in the educational platform. Vogue has also done the courses which are there in the educational platform. He has run a lot of server pipelines, and not only running the server pipelines, but it's also important to analyze and interpret the data. Both Apar and Vogue has done a very good job in that aspect. Now, these are the expectations from you as a part of this course. So we would expect 80% attendance of the online lectures. It might be so that once in a while you are kind of not in a position to attend the lecture, but that's okay. That is perfectly okay. But you have to kind of plan accordingly, since you already are aware of the dates when the lectures are going to take place, so you can plan accordingly. Once in a while it's okay, so that's not a problem. So the assignments that will be given to you, you kind of have to complete them, and you have to be a little more initiated. You have to be a little more engaged out here, and I can guarantee you this is a very interesting program. This is a different type of learning that you people are having in this part of India. This is a very prevalent concept in the US and other countries as well. So this will be a very interesting journey for all of you. So one more expectation is completion of the online courses. So I am definitely hopeful that all of you who are here, kind of here, will be completing all educational courses in the edu.t-bio.info. And also at the same time, you'll be running a lot of pipelines and analyzing your data, and definitely therefore interpreting the data as well. Okay, so I'm kind of end of my blabbering. So I hope you are you are excited. So am I. I mean, I'm very excited. I'm kind of very engrossed. So I am overwhelmed to be a part of this program, and I can see that you people are also very initiated. I can see a lot of chats going on in the WhatsApp group, and also in the in the associated chat with Zoom. A lot of questions, a lot of queries. So don't worry about the server thing. We're going to take care of it at our end, and we'll definitely be solving it. And you will be able to log on into the server and kind of play around. Do not do anything on the server right now. Just check it out whether you are able to log on or not. Since you already have a profile in the educational platform, you can go forward and explore the courses. You can go forward to explore the projects out there, the blogs, etc. And now I would kind of set the stage for any questions if you have. Now let us go to the chat and figure out if you have any questions, what sort of questions you have. Okay, so most of the questions has been answered by Shubhrajit and Apar and Mohit as well. So if you have any specific thing, you can unmute your microphone and ask me, so that you can you are also you can have a practice of doing things in the session that are going to come up. Okay, any questions? Please unmute your microphone and ask me.
Hello.
Yes, please introduce yourself.
I'm Arno Banerjee.
Hi, Arno.
Okay. I want to ask something. That there is in the educational platform, there are the courses. Do the courses have to be completed at par with the online sessions that will be conducted on Zoom? This is the number one. Number two, the projects I was going through yesterday on the educational platform, they were written something like they are coming soon, or there were some dollars, $45, and all those things.
Don't worry about that.
Okay, so we can access that.
Definitely. So are you kind of over with your questions, or do you have anything more?
No, that's all.
Okay, great. Thank you, Arno. So the first thing is that it is wise to do the educational courses, definitely, because whatever lectures, I mean, if you attend lectures in your university, you definitely go back and read books, right?
Yes.
So these are the books that we are providing.
Okay.
So it is definitely useful if you go back to and do the educational courses. And
no, I'm asking, do they have to be meaning that are they have to be done at par with the lessons that when they're happening on the Zoom, or if I wish I can
no, not at all, not at all. So Zoom is only for the lectures, for a kind of introduction.
Because sometimes it is easier to understand the lectures if I have a reading knowledge of the matter before. That's why I'm asking.
Sure, you can always go back, go to the lectures prior to the go to the educational courses prior to the lecture. That's not a problem. But it is wise to listen to the lecture first and then go to the educational courses, because in the lecture itself we will be giving the pointers where to start from, how to go forward, and what are the expectations, and what kind of things you are kind of will be doing out there. So instead of definitely you can play around in the educational platform, but while we are addressing the lectures here in the Zoom, later on you can go to the courses and complete it. So the courses are not at par with the Zoom lectures. It is at par with the entire length of the course, the 3 months course that you have registered for. Does it answer your question?
Okay, thank you so much.
And the second part is that during the entire length of the course, all the projects that you are seeing out there will be available to you. So that's you have already paid for that. So don't you worry about that.
Thank you so much.
Thank you, Arno. Anybody else?
Yeah. So
Arijit had a question that can he use his previously owned FastQ data on the platform to run?
Once again.
Could he have the access to run the FastQ files that he already owns or has?
So Arijit, I think he's a scientist at Adamas University, right, Arijit?
No, sir. I am a faculty at Vidyasagar College. I'm
Yes.
Arijit.
Hi, Arijit.
Okay. So, as per my PhD program, I do have some first Q data with me.
Okay.
So, am I allowed to process that in the server?
We have to kind of look at the data first of all, okay. Because in the server itself, since there are a lot of already uploaded data out there, during the time of the project, you can definitely do it. But before actually analyzing the data, which is there for yourself, you have to understand how the platform runs. What
definitely,
are the
definitely. I want to learn that particular thing only.
Exactly. So, instead of analyzing your own data in the beginning, it would be wise that if you go through the platform step by step, and then you can, obviously you can try to, analyze your own data.
Okay.
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 Great to have you
Definitely.
here.
Thank you so much. Thank you so much.
Yeah. Thank you. Anybody else? Any question you have? Please unmute your microphone and ask me. Okay, Shreyoshi, yeah. So profile groups, Amity University program. So which profile you are talking about?
They are having issues with viewing the progress and assignment tab in their profiles.
Okay, your own profile. So they have already made it.
The profiles, to access the profile, I would like to say that go to the profiles tab of yours on the top right corner.
Let us do it apart.
Sure. Could you open the screen?
Yes, I will do it. Okay, so if you go down, you can find, yeah, please, Apar, go on.
Yeah, could you scroll up?
Sorry, I'm going to the home.
No issues. Go to your profile picture.
Okay.
Go to profiles tab.
Okay. Yes.
Go to groups.
Yes.
Here you can see Paul Joy joined to the group Amity
Yes.
University Bioinformatics.
Yes.
Click on Amity University Bioinformatics.
Yeah.
So from here you can see the assignments, curriculum, progress bar.
Yes. So the best way to go out here is to, to kind of go to the organizational page, to go via this. So if you go to the organizations, and there are a list of organizations out here. So if you can click there, you come directly to this page. So instead of kind of going to the profile, you can always do it, but that's a roundabout way. The best way to go to the organization page, we have created organization page for that purpose only. For example, we have an LBRN organization page. We have a bioinformatics for precision oncology. Let's go here and try to check out how does it look. For example, I'm a part of this precision oncology program. So is Apar, Mohit, and Sahil, all these people. So it's taking some time. Yes, as you can see, it looks the same, only the picture differs and the description differs. So if you go to your profile, to the university organizational page, particularly to your Amity University, you can find this page. So each of your profile, for example, the visual that I can see out here is for me. So most of you will be seeing this thing only. So here you have a forum, You have progress, the kind of progress that you are making. You have the curriculum out here. I showed this thing to you again. I'll show once again. So if you click on the curriculum tab, so you can see what all will be covered in the entire length of the 3 months program.
Yeah, Pratim, I mean, they are not been able to see in this, if you go back, couple of things they are not been able to see with that. One is assignment and another is progress. So that's fine. We'll get back to you.
No, but progress, there will be none. Progress, in the progress, there will be none because they have not made any progress as yet. As I was telling you in the progress bar, this is the progress that is shown of me. This is my progress. This is my progress. Okay. You will know. Are you being able to see this graph? Are you able to see this graph, Shreyoshi?
No, sir, the entire tab is unavailable. For
The entire tab is on. Okay. So this progress is not there.
Yes, sir. This progress and assignment tab both are missing from the, the place. Both of these are not there.
Okay.
I think, Pratim, these are
Okay.
for faculties to see the progress for incomplete. Every user can see their progress from their points and stuff. So, that's how it is. And I think
Yeah, maybe so. Yes, maybe so. Yes, maybe so. So you will be able to see home, forum, curriculum, and courses.
Yeah. So we have exceeded time.
So don't worry. If you are not being able to see it, I think that's not a big deal.